Hello everyone, welcome to my channel Biochemistry by Dr. Tejas Shah. Today we will see about the metabolism of serine and threonine. So first let us see some of the key points regarding these two amino acids. So as I mentioned, these two are hydroxy containing amino acids. They both are optically active. Here you can see serine have asymmetric carbon atom and threonine have two asymmetric carbon atom. So they are optically active. Both contains uncharged polar side chains. Serine is non-essential amino acids, so it is synthesized in our body, and threonine is essential amino acid, so it must be provided in the diet. Serine is glucogenic amino acids, while threonine predominantly glucogenic amino acid, but partially it is considered ketogenic. Why it is so? Then let us see in the discussion. First we see about the serine. So serine synthesis. So it is formed from phosphoglycerate, that is the intermediate from the glycolytic pathway, and this is the major source of the serine. Next is the glycine through the hydroxymethyl transferase reaction that we have already seen under the glycine metabolism. Transamination of hydroxypyruvate with the alanine. So we see the major source how the serine is synthesized into the reaction in the next slide. So here you can see under the glycolysis the first intermediate, that is 3-phosphoglycerate, is formed. It is further oxidized by the enzyme dehydrogenase to form 3-phosphohydroxypyruvate. Then it undergo transamination reaction with the glutamate to form phosphoserine and alpha ketoglutarate, and here the pyridoxal phosphate acts as a coenzyme. Next is phosphoserine is acted upon by the phosphatase, where the inorganic phosphate is removed and there is a formation of the serine. Now once the serine is formed, its level is sufficiently reached, it represses the enzyme dehydrogenase, and as well as it will inhibit the phosphatase enzyme. So this is how its synthesis is regulated. Now if we see the degradation of the serine, it is first degraded to form pyruvate by the serine dehydratase enzyme. Here you can see in the reaction, serine first undergo dehydration to form amino acid, the intermediate, and it is removal of the ammonia group. There is formation of the pyruvate, and thus it enters into the glucogenic pathway. Next is it will form glycine through the hydroxymethyl transferase reaction. So serine hydroxymethyl transferase reaction to form glycine, and glycine is further undergo the degradation by the glycine cleavage system. And third, of the hydroxypyruvate with the alanine. So how it takes place, let us see. If you see in this reaction, serine is transaminated with the help of pyruvate to form alanine and hydroxypyruvate. Here pyridoxal phosphate acts as a coenzyme. Then hydroxypyruvate is reduced to form the glycerate, and the glycerate is receive the phosphate group at the second position to form the 2-phosphoglycerate, and it enters into the glucogenic pathway. Now if we see the metabolic function of the serine, it helps in the 1-carbon metabolism, that is formation of the methylene tetrahydrofolate. Next it helps in the formation of the cysteine, and this is the reaction by which the cysteine is formed. Serine first combined with the homocysteine by the enzyme cystathionine beta synthase, and there is formation of the cystathionine. Then cystathionine is converted to form cysteine and homoserine is upon acted by the lyase enzyme removal of the ammonia group to form alpha ketobutyrate. So here you can see there is formation of the cysteine. Next is the choline synthesis. It is also called the glycine serine choline cycle. Here you can see in this reaction serine is decarboxylated to form ethanolamine, which receives first methyl group to form methyl ethanolamine. Again it receives the methyl group to form dimethyl ethanolamine. Again it receives the methyl from the 1-carbon pool, and it is formed the choline, that is trimethyl ethanolamine. And this choline helps in the synthesis of the acetylcholine, that is the important neurotransmitter. Then choline is converted to the betaine. Betaine is also called trimethylglycine. So here you can see the glycine acts as a methyl group salvage. So betaine upon demethylation to form dimethylglycine, and it is further converted to the sarcosine, that is the methylglycine, and again it will form the glycine. Next we see there is serine helps in the formation of the alanine, that is dehydration followed by the transamination, already seen under the degradation of the serine. Next, serine helps in the synthesis of phospholipid like phosphatidylserine, as well as the sphingosine. It also contains the serine, and sphingosine is further incorporated to form sphingomyelin. So serine analogs can be used in the drug therapy. For example, azaserine is an anti-cancer drug and cycloserine is an anti-tuberculous drug. Serine also helps in the regulation of the enzymatic activity through the covalent modification, that is helps in the protein phosphorylation. It also forms the active catalytic residue of some of the enzymes that are called the serine proteases. So the serine protease inhibitor, the clinical significance lies with the deficiency of serine protease inhibitors, so it will cause the emphysema. Next is serine helps in the glycoprotein synthesis. The sugar groups are attached to the hydroxyl group of the serine or threonine, and it also helps in the synthesis of the selenocysteine, that is considered as a 21st amino acid. Now we see the threonine. Its metabolic function is it helps in the synthesis of glycoproteins, where the sugar group combines with the hydroxyl group of the threonine. It is same as that of the serine. So both serine and threonine helps in the synthesis of the glycoproteins. Now if we see the degradation of the threonine, the first major pathway, that is by the threonine dehydratase reaction, where threonine is converted to alpha ketobutyrate, where ammonia is liberated. Then alpha ketobutyrate undergo oxidative decarboxylation to form propionyl coenzyme A, which is carboxylated to form methyl manonyl coenzyme A, and it is further converted to the succinyl coenzyme vitamin B12 as a cofactor, and then succinyl coenzyme A enters into the TCA cycle. Thus it helps in the glucogenic pathway. If you see this reaction by propionyl coenzyme with the TCA cycle, it is also reflected there in the odd chain fatty acid oxidation. Usually fatty acids cannot be converted back to the glucose, but this is one exception where the odd chain fatty acids are considered as a glucogenic. Now the second reaction by which threonine is catabolized, that is by the threonine aldolase enzyme, that is the minor pathway, where it will form the glycine, and glycine is further undergo the cleavage system degradation. Then also formation of the acetaldehyde. Acetaldehyde further oxidized by the enzyme dehydrogenase to form acetic acid, and it will receive the coenzyme A by the enzyme thiokinase, which requires ATP along with the magnesium ion, and it is converted to the acetyl coenzyme A. So forming the acetyl coenzyme A, it is considered as a ketogenic also. So by this pathway you can say the threonine is the glucogenic, and this is the major pathway. So predominantly threonine is glucogenic, and by this pathway it is partially considered as a ketogenic. So these are my references for this presentation. Thank you. Please like, share and subscribe to my channel. Click the bell button to get all the notifications. Thank you all.